Welcome to part 2 of the lecture on plant growth and development. Please ensure that you watch part 1, plant growth and development, before watching this lecture. Please refer to chapter 16 for the recommended reading from the Tays and Zeiger plant physiology book. This subject is a component of the agricultural and viticultural degree programs offered at NMIT. My name is Dr. Nikki Cooley. In part 2 we will examine the growth of meristemic tissues. We will look into some detail about growth of root apical meristem, the root structure, the interactions with root growth and auxin, and interactions with root growth and cytokinin. We will then go on to look at shoot apical meristem. We will examine leaf geometry and patterning and lateral root growth. The root tip has 4 developmental zones. In zone 1, the root cap. This contains a unique set of initials that covers and protects the apical meristem. It also functions in the perception of gravity, also called gravitropism, and the secretion of compounds. In the developmental zone 2, the meristematic zone lies just under the root cap. This contains the initials that divide and differentiate into various tissues of the root structure. In the third zone lies the elongation zone. This is the site of rapid and extensive cell elongation. And finally, the maturation zone. This is the region where cells become differentiated after division and elongation has ceased. Organs such as the lateral roots and root hairs may form at this stage. The figure on the slide shows a visual representation of the root tip structure. You will note the lateral root prydomium, the mature zone, the elongation zone, the meristematic zone, and the root cap. All of the important components of each of these zones are illustrated. The origin of the root tissues can be traced back to specific initial cells in the root apical meristem. At the center of this region is the quiescent center, or QC, named for its low rate of cell division. 4 distinct sets of initials can be defined in terms of their position and the tissues they produce. The Columbia initials, the epidermal lateral root cap initials, the cortical endoderm initials, and the steal initials. The Columbia initials divide to form the central portion of the root cap cells. Epidermal lateral root initials divide to form lateral root cap cells and epidermis. The cortical endoderm initials form the cortex cells and the endodermal layers, while the steal initials form the vascular system and the pericycle cells. The figure on your screen shows the root initials. On the left-hand side, you have an image taken from a root tip. On the right-hand side, you have a schematic drawing of where all the initial components are located. Let us talk about auxin and the role it plays in the root apical meristem. It is believed to play an important role in positioning of the root apical meristem and guiding its behavior. The position of the QC coincides with a maximum concentration of auxin. Models show that auxin moves down from the shoot through central regions of the seedling and concentrates in the QC. Auxin is then transported outward, then inward towards the shoot. The figure on the slide summarizes some of the experimental evidence demonstrating auxin movement. This has been done by combinations of method, looking at pin protein distribution, modeling of auxin flow, and DR5 to GFP expression. Studies suggest that cytokinin acts in opposition to auxin. Auxin is synthesized in the shoot and transported to the root, while cytokinin is synthesized in the root and transported to the shoot. Auxin promotes root development while cytokinin suppresses roots while promoting shoot development. Cytokinin begins early in the hypothallus of the glandular embryo. On division of the hypothallus, cytokine expression is lost in the basal cell but retained in the apical cell, which divides further to form the QC. The following image is an SEM of a shoot apical meristem. This shows the very early stages of initiation. You can see regions P1 through to P4. Now I'm going to talk a little bit about zones and layers of the shoot apical meristem. The central zone, or CZ for short, contains a cluster of infrequently dividing cells, similar to the cells of the quiescent center. The peripheral zone, or PZ, divide more frequently to produce cells of lateral organs such as leaves, while the rib zone, or RZ for short, contains cells that give rise to the internal tissues of the stem. Shows a high-power microscopy of the zones and the layers of the SAM, the shoot apical meristem. As we described in the previous slide, you have the central zone where the cluster of infrequently dividing cells occurs. The peripheral zones, either side of the cluster zone, and the rib zone. These cells contain and give rise to internal tissues of the stem. The shoot apical meristem also has cell layers that contribute to specific tissues of the shoot. The outermost L1 layer, this is where the epidermal layer is created. Cell divisions are anticlinal in this layer. The L2 layer is the subepidermal layer. Again, cell divisions are anticlinal. While in the inner L3 layer, internal tissue is generated. Random orientation of cell division occurs in this layer. You can see the following slide demonstrates the locations of the L1, L2 and L3 layer. Anticlinal division, while we have random orientation. This helps with the shape of the stem. The architecture of the shoot mainly depends on the production of the lateral organs, leaves and branches. The root systems also increase in complexity by the outgrowth of the lateral roots. This is collectively known as vegetation orgogenesis. Now we shall look at leaf initiation. The characteristic arrangement of leaves on the shoot is termed phytolaxy. Three basic phytotactic patterns are linked to the pattern of initiation of leaf primordia on the shoot and the apical meristem. There's alternate, dissociate, and spiral. It has been known for some time that leaf primordia initiate where auxin accumulates. Here we have a visual representation of the components of the shoot of the leaf initiation, characterizing auxin movement. Leaf geometry can be described by a proxial distal axis, base to tip, a lateral axis, that is from one edge of the leaf to the other, and an abaxial to adaxial axes, which is the upper to the lower surface. Mechanisms by which leaf patterning occurs remain unclear, although several important genes have been identified. The phytotastic mutant fails to establish leaf patterning, and the leaf forms a cylindrical shape. Here we show a visual representation of this mutant. We start with the wild type that has both adaxial and abaxial. This leads to inductive interactions between the distinct layers. Lateral growth follows, and you can see abaxial and adaxial growth. While in the mutant you only get abaxial growth, and this results in a filamentous shape as opposed to a laminar shape. Now we will examine lateral root growth in more detail. The first signs of lateral root initiation are periclinal divisions of a small number of cells in the pericycle. The new root tip eventually ruptures and the endoderm and cortical layers of the parent root. A new root apical meristem is established at the tip of the lateral root. Lateral root patterning is influenced by auxin concentrations as well as environmental factors such as nutrient availability and soil moisture. On the slide you will see a visual representation of root development in the number of processes that we have described. These can be separated into 5 distinct stages. In the next series of lectures we will look at plant hormones and their interactions. I am going to just touch very briefly on growth and development and hormones in this slide. This image is from a paper, Phytochromes Regulate All Aspects of Plant Growth and Development. It is from a paper published in Nature by the authors Santer, Iran, Cardon Vascular and Estelle. It shows you where the plant's hormones are regulated with respect to the plant. The plant that they're showing here is Arabidopsis thaliana. As you can see, auxins, gibberellins, ethylene, jasmic acid, boracindole, salicylic acid, stringophone and abscisic acid and cytokinins are all involved in growth and development. The table on the slide is a comparative account of plant growth regulators on plant development. On the left-hand column you will see listed the stages of growth and development, from root development through to dormancy. The following columns after this represent whether the hormone is present with a cross or absent with a slash. The hormones that are examined in this comparison are auxin, gibberellin, cytokines, abscisic acid and ethylene. Please understand that a couple are missing from this diagram, but it does allow a very simple and quick summary of which hormones are involved at which stages of plant development. And this brings us to an end on our lecture on plant growth and development. Hopefully, after watching both part 1 and part 2, you will have an understanding of the major phases of saprophyte development. You will understand the major phases of embryogenesis and the role of apical basal polarity, and understood how auxin influences embryogenesis and movement. In part 2 we examined 2 meristemic tissues in detail, the root apical meristem, where we studied root structure, the role of the hormones auxin and cytokinin. We also looked at the growth of shoot apical meristems. We have explored leaf geometry and patterning, as well as lateral root growth. And we ended with a summary of the role of hormones in plant growth and development, which we will expand in the following lectures on plant hormones. Thank you for your time.